Xtalks connects professionals in the life science, medical device, and food industries with useful content like webinars, job openings, articles, and virtual meetings to help you succeed in your career. This life science focused podcast brings together some of our editorial staff to share insights into the latest B2B industry news to keep you up to date.
Hello and welcome to the Xtalks Life Science Podcast. I'm Aisha Rashid, Senior Life Science Journalist at Xtalks.com, and this week I'm delighted to introduce a very special guest on the show today, Dr. David Young, who is the founder and president of R&D at Processa Pharmaceuticals, a clinical stage pharmaceutical company dedicated to developing products that improve survival and quality of life for patients with unmet medical needs. This includes personalized approaches to cancer treatments with a focus on the development of next-generation chemotherapy drugs to improve the safety and efficacy of cancer therapies. Processa recently released an update on its clinical cancer program, which we will hear about from Dr. Young. Dr. David Young is Processa's initial founder, president of R&D, and former president and CEO. He has over 30 years of pharmaceutical research, drug development, and corporate experience. He has served on the board of directors and as chief scientific officer of Questcor Pharmaceuticals from 2009 to 2014. Dr. Young has expertise in small molecule and protein non-clinical and clinical drug development. He has also served on FDA Advisory Committees, was co-principal investigator on an FDA-funded clinical pharmacology contract, has been a key team member on more than 30 NDA supplemental NDA approvals, and has served on NIH grant review committees, among his many other accomplishments. Dr. Young received his Pharm D-PhD with emphasis in Pharmacokinetics and Pharmaceutical Sciences from the University of Southern California. To hear more about Dr. Young's work in personalized next-generation chemotherapy drug development at Processa, let's hear from him now. Welcome to the show, Dr. Young.
Hello, thanks for the invitation.
So, Dr. Young, can you start off by telling us a bit more about Processa Pharmaceuticals' missions and goals, as well as when, how, and why you founded the company?
Sure. We founded the company in about 2017, 2018. And we did it because we saw many patients of all different kinds of conditions needing other therapies, whether they had no therapy or they had therapy that was below what they needed. And so it really was an issue of finding patients or finding drugs which could treat patients with unmet medical needs. We then changed over in the beginning of this year to just approach patients who have oncology unmet medical needs. So now we're focusing just on the oncology area for patients who need other treatment options that they have right now.
Excellent. And so could you tell us a bit more about your journey to founding Processa and what led you to that?
Sure. Well, it all actually, I'm not going to go way back. I'm just going to start from the more recent and then step back a few steps. So more recently, I was on the board of directors of Questcor Pharmaceuticals. We had a drug that was to treat infants who had infantile spasms. Okay. And while I was on the board, we realized that more patients needed this drug than we thought. And we realized also we needed to get FDA approval for this drug. I then moved from the board to the management team to try to get FDA approval. And in 12 months after moving to the management team, I got FDA approval with my team that I brought in for infantile spasms. That approval allowed many more patients to receive this drug. Infantile spasms is a condition where about 50% of the patients used to die from it, the babies. Okay. The other 50% became so mentally disabled or physically disabled that it was very, very difficult to take care of the patients. Quality of life was bad for the patients, wow. etc. So our drug, which was already approved on the market for something else, was used off-label for infantile spasms. We needed to get it on label so more patients could get the drug. And so that's what I was brought in to do. My team, we negotiated things with FDA. We used our regulatory science approach, which I'll talk briefly about in a second. And then we were able to get that drug approved in less than a year. And we got additional 17 other indications approved beyond that. Right? So that changed the company from a market cap of maybe $10 million to when we sold it a few years later for $5.7 billion. So it was very good.
Wow.
Very good for the patients, very good for investors, very good for Questcor. Now, how did I get there? I got there because when I used to be on the faculty of the University of Maryland in the 90s, FDA came to me and said, David, we know what you can do. Can you help us to define and develop what we call regulatory science? And so when they came to us to do that, I said, okay, sure. And let's get a definition of it. So we started to define what really regulatory science was. And FDA said, what we need clinical science to back up our regulatory decisions. So I said, okay, I can do that easily. So they started defining regulatory decisions or problems they had. I would define the clinical studies to support those decisions and then do the clinical study work and get the data and then support the decisions. In addition to that, at the same time, FDA said, well, why don't you help our reviewers think through the process of regulatory science? So I actually had meetings with the FDA reviewers, and they would bring cases to me of things they were doing because I had clearance, FDA clearance. Mm-hmm. And they would say, here's the drug, this is the issues, how should I look at it? Well, I didn't know anything. It was brought to me that minute. I didn't know anything about the drug. I didn't read about the drug. And so I would try to figure out how they're thinking, right? Why are they thinking a certain way? Why are they thinking the science is saying this? Why are they thinking the science is saying that? And why is that helping them to make a regulatory decision? And I would guide them in their thinking process. So I learned what the process needs to be in order to make regulatory decisions that way. And I wasn't an FDA reviewer. I was just somebody who was helping teach them how to do things. That's where everything, I won't say blew up, but that's when everything expanded into us my team really doing regulatory science, and then moving into the pharma industry and then building our regulatory science with over 30 FDA approvals and indications across all of FDA. Mm-hmm. And we use that regulatory science approach to help us get through and help guide people through these different drugs.
Wow, that's excellent. And because you don't really, there's often there shouldn't be, but there's often, a disconnect between pharma companies or even academic centers trying to get approvals. And there's a disconnect in terms of the FDA trying to understand the side they're on and vice versa. So that's really great that you were able to become a bridge of sorts between the two areas there, essentially. So that's really great to learn about. And so I know that you mentioned that Processa has shifted its focus now into oncology. And so could you tell us about Processa's next generation chemotherapy drugs in terms of how they're designed to be more personalized and offer improved safety and efficacy?
Sure. When we started developing the company as an unmet medical need company, people would knock on my door, call me, send me emails saying, I have this drug. You might be interested in this drug, David. And so we reviewed about 50 or 60 drugs to say which drugs we wanted to bring in. Mm-hmm. So we brought in a couple of drugs that are not oncology, and then we brought in 3 oncology drugs. The reason we brought these 3 oncology drugs in is because we thought these drugs are a little de-risked. And the reason they're a little de-risked from the typical oncology drug is because the active molecule of these oncology drugs was the same active molecule that's in an existing FDA-approved drug already. And all we were going to do is tweak it, do something so it's metabolized or distributed differently when it gets into the body. We're not changing the absorption. We're not changing. What we're doing is when it gets into the body, we're changing it so it distributes more to the tumor and or less metabolism occurs to something that might cause side effects. Or maybe it's metabolized more to something that actually kills cancer cells. So we in a sense, we were modifying the internal systemic working and how the body treated that drug, how the body handled the drug. That's what we're doing. Each drug, each of our three drugs, it's a different approach for each three. The general theme is the same. We're changing the metabolism and or distribution, but how we change it could be different with each drug.
Oh, okay. Nice. And so could you tell us a bit about the three drugs that are in your pipeline currently?
Sure. So we have one drug called next generation capecitabine. Capecitabine was developed by Roche, and we actually were involved in part of that development. And the capecitabine drug is actually a prodrug of 5-FU. 5-FU, which is 5-fluorouracil, was a drug developed in the 1950s by Charles Heidelberger. Right? When he developed it, it was one of the first cancer treatment drugs around. In the late 1970s, I actually worked on 5-FU in patients for my dissertation, for my PhD. And Charles Heidelberger was on my committee.
Oh, cool.
Yeah. So, and he would tell me sometimes, David, we got side effects. How can you decrease the side effects? And I would look at him and say, I don't know. I'm a graduate student. You're the one who has the doctorate degrees. You should know. I'm just a graduate student. But it kind of stuck in my head. How do you decrease the side effects? How do you increase the efficacy?
Yeah.
And so when this next generation capecitabine drug or drugs, because it's a combination drug, came across our desk, I said, this is interesting. This is exactly what Charles Heidelberger asked. How do we decrease the side effects and increase the efficacy?
Coming full circle.
Exactly. And I said, gosh, it looks like this combination of two drugs could do it. And so that's why we chose that one. Okay. The other two are, again, very similar. These are the active molecule is already proven to kill cancer cells. And what we've done is, in one of them, next generation gemcitabine, we've changed it so this other molecule, we tweaked the molecule a little bit, and it's now metabolized more to an active molecule than gemcitabine. Good, get less resistance, etc. The third drug is next generation irinotecan, and the next generation irinotecan drug, what happened is we put on another molecule on the drug. What's administered in the body, that molecule helps it to go into the membrane of cancer cells more than normal cells.
Okay.
The active molecule, once it gets in the membrane with this other molecule hooked to it, breaks off from that other molecule and then it goes into the cancer cell. So it's kind of specifically distributed to the cancer cell more.
Right.
Again, we're talking about metabolism, we're talking about distribution changes, which will result in better safety and better efficacy.
Very nice. Very nice. And, we hear about targeted approaches in other areas like immunotherapies and things like that, which is really big. But to take existing drugs and to modify them, I think that's a huge area. And it's really great that people like yourselves have been working on that to get next generation chemo, because we know chemo works, but right. So why not take that and make it better? So
that's it. And that's the key. Yeah. Make it better. And one of the things we have to realize is that when we're talking about the next generation chemotherapy, we're talking about something that patients need. Yeah. Because, it's not a brand new target. I'm not searching for a brand new target. I'm not taking a given molecule and making a brand new molecule out of it and to hit something differently. I'm taking what's already there as the active molecule to kill the cancer cells. We know it kills it. So if I can get more into the cancer, it'll kill more cancer. If I can form less metabolites that cause side effects, I'll have less side effects. All can increase not only the number of patients who receive the drug, because a lot of these patients, in chemotherapy, they'll receive the drug, and then what happens to the patients is they have to either decrease their dose because of side effects, or they have to stop taking the drug because of side effects. So if I can get rid of those side effects, maybe those patients can have optimal therapy. Right? And that's what we're trying to do.
That's really exciting. This really opens up like a lot of new avenues, I think, and I think
Correct.
it needs to be talked about a lot more and be put out there. So this novel approach, and so actually, this kind of maybe goes into my next question: what sets processes, technologies, and approaches apart from others in the field in terms of? I guess I'm not sure if you can go into the exact specifics in terms of what molecules you're combining and things like that, but as
Yeah.
you're aware of other competitors, what sets Processa apart?
Yeah. So I can't go into that
Yeah,
the changes on the molecules and things like that.
it's
But what I
proprietary,
can tell,
definitely.
yeah, what I can tell you is that everything we do follows the regulatory science approach.
Mm-hmm.
So what FDA does right now is when you submit an application for approval, they'll look at the benefit risk.
Yeah.
And they'll say, is the benefit outweigh the risk? And if it does, I'll give the drug approval. Okay. And what does outweigh mean? For each drug, it's different.
Yeah.
And for each disease, it's different. All right. But they'll evaluate the benefit versus risk. So what we're doing is we're trying to increase the benefit, decrease the risk, increase efficacy, decrease safety, or improve the safety. Right? And if we can do that, then the probability is number one, we have greater chance of approval because it's already a molecule that kills cancer cells. We know that. Number two, guess what? Compared to the existing drug, this is better benefit and lower risk. Who's going to use the other old drug? Nobody.
Right.
Everybody's going to use a new drug, right?
Right.
So it can also commercially take over the market.
Absolutely.
And then the third, of course, is the patient, which is the most important. It really can help the patient make a better quality of life, treat more patients on optimal regimens, et cetera, et cetera. So that's kind of our idea.
Definitely. And so from a regulatory perspective, when you have a drug, I mean, this is obviously different because you're developing a next gen drug, making modifications, but we hear about repurposing drugs, this is kind of playing on that idea, but taking it further, of course. And so from a regulatory perspective, is it easier to convince the FDA or to get them on board? Are the timelines a bit shorter, or how does the regulatory aspect work for something like what you're doing at Processa?
A good question. That's a very good question. There's multiple aspects that the FDA has to look at. The first is, does the mechanism of action, does it really kill a cancer cell, right? And what's the mechanism of hurting other cells? So that's the first aspect, right? And when the agency looks at ours, they will look at ours and say, well, we know it kills cancer cells because it's already approved. So that box is checked.
Right.
They don't care about that one. The next question is, well, does it really actually improve the benefit risk, improve efficacy, improve the safety profile? We still have to go through the same hoops to do the exact same thing to prove clinically it's better. Okay. And again, those hoops are required by FDA. Again, those hoops are required in order to commercialize the drug. So we have to do that. It's not any faster because we have to do the same studies. It's not any quicker, anything like that. The other thing we have to remember too is more recently, FDA put out what's called Project Optimus Initiative, okay, which is how do you determine the optimal dosage regimen for an oncology drug? And they're using a different approach than what we used previously. They're using the approach that we've been using with all other non-cancer drugs for a long time, which is actually looking at the exposure versus safety relationship, looking at the exposure versus efficacy relationship, and then determining from that, which is the best dosage regimen to go into my studies, which is the best dosage regimen to give to my patients, which is the best dosage regimen to commercialize. That in itself has added more hurdles for us to jump through. It's not our molecules, it's that process that has required us now to jump through more hoops. But guess what? I believe that those hoops will be better for the patient. So, because they're more optimal. Yeah, so you might have less side effects, but the same efficacy. That would be great, right? Better quality of life. So, it's very rational to do it. But there are more hoops now, but it's not because we're the next generation drug. It's because FDA has appropriately said we have to do some extra steps.
Makes sense. And so I know that Processa recently released some results from its latest interim analysis of, I think, one of the compounds. I think the first one that you mentioned. So could you talk to us a bit more about that?
Yes. So if you think of, I think you mentioned it in your introduction. If you think about what's happening now in cancer, a lot of it is looking at the genetic makeup of the patient, immunotherapy, et cetera. Okay. And the reason they do that, they say, if you have this general genetic makeup, then I can give you this regimen or this drug. If you have a different genetic makeup, I shouldn't give you that drug. It's not going to work as well, or I got to give you a different regimen. Okay. I could take that a little further. Back in the late 70s and early 80s, when I was a Pharm D-PhD student, we did what was called therapeutic drug monitoring. So what we did is not for cancer. What we did was for some other drugs, we would give the drug to the patient, and at some point in time, we would measure how fast that drug's eliminated, what's happening with that drug, which is called the pharmacokinetics. We would measure it, how it's being metabolized, how it's being distributed. We would kind of get a handle. And you, for example, might have the drug in your body for 10 hours. Me, I might have the drug in my body for 24 hours, right? You therefore might need a different regimen. You might need the drug more often. Well, I don't need it as often, all right? So the individualization was developed long ago with other drugs through therapeutic drug monitoring. What we have found is that for our next generation capecitabine, there may be that approach that one needs to take. You might need a dose sooner or higher dose in order to get efficacy. Well, I don't need it. And we might be able to do that. And I say might, it hasn't been proven. It's just, we found that this is something that might be occurring. I may need it less, you may need it more. So we should individualize our dosing regimen by measuring what's going on. That's the idea. Now, that's different than the genetic approach. The genetic approach is everybody with the same gene gets the same dosage regimen, right? This is actually individualize it by personalize it.
Right.
So if we can do it, and if in fact it applies to the different patients and makes your therapy better, it's worth it.
Absolutely. Yeah, that's great. And so let's see. Right. Just going back to, I think, capecitabine, was that the drug that you had results for the latest interim analysis?
Yes, next generation capecitabine.
Right. Okay. And what can we learn about that so far?
Well, what we really found so far is there is a possibility, and I'm not saying it's definite yet. There's a possibility that we might be able to individualize the therapy.
Okay, so the dosing regimens were
yes, we might be able to individualize the dosage regimen.
Awesome.
And we might. I'll emphasize a possibility.
Right.
It's not done. It's just a possibility.
Right. That's how we operate in science, right?
Yes, exactly
could,
right.
should. Nothing is definitive until,
Yes, right. Until you get the data. Until you get the results and prove it.
Data. Exactly. And so I think I just want to ask you, given your, extensive expertise and experience in the field of drug development and now in oncology, I just wanted to ask, how have you seen the landscape really change in drug development? I know you hit on a lot of great and important topics from a regulatory perspective, and then also in terms of the biology of designing these next generation chemotherapy drugs. So how have you seen the landscape of drug development evolve? How do you see it evolving in the future, as well as personalized medicine? Like you mentioned, it's not all about genetics and tailoring things at a genetic level, but it goes much further, and there are so many more aspects to consider. So where do you think things have come, and where do you think things are going?
That's a good question, and I don't have a one answer to this, and I don't necessarily have the correct answer. All I can tell you is that from 1970s to now, we have seen that development has changed, both from a financial need point of view in terms of doing the studies, conducting the studies, from a patient need point of view in terms of finding other treatments that will make their life better, and from an FDA point of view. I mean, the FDA looks at drugs differently now and reviews drugs differently than they did back in the 70s and the 80s. Back in the 80s, we had meetings with the FDA. And in the 90s, we had meetings with the FDA.
Mm-hmm.
We could actually discuss the science, for example.
Right.
After COVID, we have not been having those types of meetings, unfortunately. We haven't been able. I remember back in the early 90s, I was meeting with FDA on a drug, and they brought something up about the drug. And I said, that's a good point. Maybe we should design this study a little different. And so I got on the chalkboard and I started drawing on the chalkboard how we might be able to design it differently. And they said, yeah, that's what I'm talking about. That's exactly what I'm talking about. That interaction's not there. But that interaction's not there with each other in our companies sometimes.
Right.
Yeah. So, the sooner we can get back to that interaction, both in our individual companies and our teams, as well as that interaction with the FDA, I think there's an advantage. We can accomplish more that once we get back there.
Absolutely. And so, currently, what challenges, apart from the lack of interaction, what challenges are there really from a regulatory perspective in getting a drug from preclinical development through to commercial launch? You mentioned that there are a lot more hoops, but at the end of the day, that is more beneficial for patients and commercially as well. So what regulatory aspects should people that are, new in the field or coming into the field really consider, and how have you seen the regulatory landscape change?
Yeah, from the 90s to now, I haven't seen much of a change. I think
is that a good thing or a bad thing?
Well, let me qualify much of a change.
Okay. Right. What does that mean?
When I say much of a change, I mean FDA still looks at the benefit risk profile.
Yeah.
Okay, and they still evaluate that. And they're not just evaluating it from a clinical study, they're evaluating it for the patient need.
Yeah.
What does the patient need? What is the other therapies they have? Which I think is fantastic. So that hasn't changed. That's still there, and that's good. That needs to be there. I think what has evolved over the last 20, 30 years is that companies have taken a different approach to show that benefit risk, or to bring up that benefit risk to the FDA, right? And some companies just say, I'm going to throw all the money at it and do whatever, and do everything that they want, everything that it could be possibly done, and hopefully they'll find the gem in there, and they'll find the diamond in the rough, and they'll say, I approve it. And so that's what they do. Well, that's hundreds of millions of dollars that are being thrown out to these patients. And that could be studies that don't help the patients at all. Right. So, I would prefer, if I had my way, I would prefer that companies take a more targeted approach to improving and developing drugs. Targeted, not meaning targeted patients.
Right.
Yeah. Choose studies that are going to help patients. Choose studies that help get your drug. Just don't do everything you can under the sun. Do things that are important for the patient, important for approval, which is also for the patient, right? Do those. Yep. Don't waste your time. Don't waste your money doing things just because you have money to throw at it. It just doesn't make sense to me, especially if there's patients involved. It just doesn't make sense to me.
Yeah. Well, what you've said makes absolute sense to me, and hopefully to our audiences here as well. So, Dr. Young, I wanted to thank you so much once again for being on the show. I mean, this discussion was so insightful and so very important as well. And I'm sure that our listeners will greatly benefit from all of your insights and perspectives and all of the experience that you have and your expertise. And we look forward to hearing more about Processa in the coming years and more about your work as well. So thank you once again.
Thank you very much. And I thank you very much for the invitation. I really do appreciate it. Great questions.
Take care. Bye-bye. Bye.
Thanks for listening to the Xtalks Life Science Podcast. If you enjoyed our discussions today, please share the episode with your friends and colleagues, and be sure to subscribe in order to be notified when a new episode is released. To join in on the discussion, you can find Xtalks on social media, email podcast@xtalks.com, or comment on the articles directly. Links are in the show description. Take a moment to join our community at xtalks.com to get access to everything we have to offer, including webinars, job listings, virtual meetings, articles, and more. The views and opinions expressed in the podcast are those of the speakers sharing them. They should not be taken as professional advice and do not necessarily reflect the policy or position Honeycomb Worldwide. For further information, email us at podcast@xtalks.com. Thanks for joining us, and we'll see you next week.